Okay, so now we want to talk about the counterpart to photosynthesis, that is cellular respiration. And so we see here, when we kind of look at the big picture, that these two reactions are related to each other, in that, as we see here with photosynthesis, again, we recall we're taking carbon dioxide, water, using sunlight energy, producing sugars and some oxygen. Well, in cellular respiration, and of course there's a series of reactions here, the light reactions in the Calvin cycle. Well, in cellular respiration, we're basically going to be doing the reverse reaction, but as we'll see, we're going to use different series of steps. We're going to use glycolysis, the citric acid cycle, and the electron transport chain, which we'll see that electron transport chain has some similarities with what's happening in cellular respiration. So we basically consume food produced by plants, or that comes from animals that have eaten plants, and we break those foods down, extracting energy from them, using oxygen to do that, making us an aerobic organism. That is, we do our respiration with oxygen. And so in the process of breaking those foods down, we create CO2 and water, which are the raw materials of photosynthesis. Now, the purpose of doing all this is to extract energy from those foods, and we're going to use that energy to produce a bunch of ATP, which is the molecule we use in our bodies to do work of various kinds. So our circulatory system is involved with this, because it's how the food gets around our body and how the oxygen gets around our body. And so it delivers the organic molecules, the sugars, and the oxygen to cells, and it's in those cells where we have our mitochondria that are carrying out this reaction of breaking down the food in the presence of oxygen. The CO2 is a waste product. We just release that. The water can be used in the cell or excreted from the cell if it's needed somewhere else. We generate a lot of those ATP. We also generate a fair amount of heat. Being warm-blooded animals in particular, we have a fairly high metabolism and generate quite a bit of heat. That heat is then dissipated from those cells, and then you remove from your body as well. So there's our overview. So now let's start looking at some of the details. So here's our reaction. You'll see we are basically doing redox reactions in which glucose is being oxidized. It's having electrons stripped off of it, and oxygen is being reduced. It's having electrons added. And so in a series of steps, we're going to strip electrons off of glucose, oxidize it. It's serving as the reducer, right, because it's giving up electrons. Oxygen, we'll see, will be the ultimate oxidizer, and in the process of gaining those electrons is reduced, and the oxygen we breathe in ends up in water inside of us. And the carbon that's in glucose and the other foods we eat ends up as CO2 that we breathe out. Now, in cellular respiration, we saw an electron carrier called NADPH, which was produced in the light reactions and used in the Calvin cycle. Well, here we have a couple other different types of electron carriers, one called NADH and one called FADH2. And we'll see how we're going to be reducing these molecules. We're going to be adding electrons to them to create these electron carriers that are going to play an important step in the overall process here. NADH and FADH2, NADAs and FADAs. All right. So now the overall process of cellular respiration, that is, respiration with oxygen, has several steps. First is glycolysis, and it's where we're going to take the glucose, this 6-carbon molecule, and break it down into 2 3-carbon pyruvates. In the process, we're going to make a little bit of ATP, and we're going to make a couple of these electron carriers, these NADHs. And as we see here, they are taking electrons with them over to the electron transport chain. Now the pyruvates are going to be modified in this, what's called this preparatory reaction or phase. And the process of being modified, we're going to produce some more NADH. And then the modified pyruvate, which is this molecule called acetyl-CoA, will enter the citric acid cycle or the Krebs cycle. And there we're going to complete the breakdown or oxidation of the original carbons that were in the glucose. We're going to generate a little bit more ATP. We're going to generate a lot of these NADAs and VADAs, these electron carriers. And in both this step and the citric acid cycle, we're going to produce carbon dioxide. We're going to produce 6 to be exact, because there are 6 carbons in glucose. Now over the electron transport chain, we'll see we're going to use all these electron carriers, and we're going to, they've gained electrons in the earlier steps. Now we're going to take those electrons off, and we're going to use the energy from those, stripping those electrons off, to produce a whole bunch of ATPs. You'll see it's in this last step where we produce the majority of the ATP. Okay, a little more for this video. Alright, so now we're outside the mitochondrion, and that's where glycolysis happens. Glycolysis is a process that all organisms share. It's the first step of breaking down organic molecules to extract the energy from them. And this can happen in 2 ways, or we can divide organisms into 2 kinds. You can have organisms who use O2 and organisms that do not use O2. That is, those that are aerobic and those that are anaerobic. In either case, it's preceded by, or it starts with glycolysis. And glycolysis is where we take this sugar, this organic molecule, and we break it down into pyruvate or pyruvic acid, as we saw in the previous section. Now, if you have oxygen, again, as we saw, the rest of the process is going to occur just outside and in the mitochondrion. If you're anaerobic, you don't make use of mitochondria, or you don't even have them, and you're doing fermentation. So these anaerobic respiration and fermentation are sort of somewhat synonymous terms. We'll see later how there's 2 kinds of fermentation. Alright, so glycolysis. Here's sort of the nitty-gritty details of glycolysis. And this is an image from your textbook. Sorry, it's a little blurry here. So here, we're taking our glucose with 6 carbons, and in a series of steps, we are breaking it down into 2 pyruvates. Alright. Notice that we have to make use of a couple ATPs to get this process started. This is what's sort of known as the energy investment steps, where we sort of energize the system, and then we quickly break it down into 2 different molecules. Ooh, G3P, we saw that in cellular respiration, glyceraldehyde 3-phosphate, and then finally into 2 pyruvates. Now along the way, again, we're going to make some NADH molecules. We're going to make more ATPs. In fact, down here in the energy harvesting steps, we're going to make a total of 4 ATPs. We use 2 to get started, so we have a net gain in glycolysis of 2 ATPs. Whoops, 2 ATPs. All right. we're also going to produce, again, a couple NADHs. Again, all this is happening outside the mitochondrion. And again, we put a couple ATPs in. We get some NADHs out. And we get 4 ATPs out for our net of 2. Again, in the conversion of these sugars into pyruvate or pyruvic acid. Okay. So again, here's sort of our overall balance sheet for glycolysis. We've extracted a little bit of energy from the glucose. It's now in the form of the ATP molecule. Some of the energy has been transferred to NADHs, and a lot of the energy is still inside the pyruvates, and it's in the Krebs cycle and electron transport chain where we're going to extract the rest of that energy. Okay, thanks.